Hello, I would like to thank the organizers for giving me the opportunity to present my work. I am Heba Sailem and I'm a research fellow at the University of Oxford. My research focuses on studying and analyzing large-scale microscopy data. So today I will talk about ShapoGraphy, a new tool that we created that not only allow the users to visualize their data, but also to create their own visual encoding by a flexible glyph-based approach. It can be applied to any data type, but I will focus on using ShapoGraphy for microscopy data. Microscopy data is highly versatile and it can have specific challenges when it comes to data visualization. Because we have multiple, we can capture biological systems at different scales from the molecular level to subcellular and tissue to organism level. We also have this hierarchical nature. So the cells have the nucleus in them and the proteins, and then they are within the tissue, and this is within the organ, etc. They also have the properties such as texture and intensity, as well as the spatial nature of these data. So it's really challenging to develop ways that allow generalizable visualization of these data types. So in general, the existing visualization methods are very limited. So we have mostly heat maps, bar charts, and that's used with any data type. There is not only they are not intuitive and difficult to link to the original data, but also they discard the structural and the semantics of the images. So during my PhD, I devised a tool called Phenoplot, which basically aimed to facilitate the interpretation of the data by creating structures that look like what we are measuring in the first place. So I used a combination of geometrical shapes, line styles, symbols, and color hues. And you can see here, like we can, I map the data to these ellipses, which represent the cell and the nucleus. And I fill the ellipse with the symbols depending on the variable value. So this can represent protein abundance or organelle abundance. The border of the cell can represent how much cell-to-cell contact, so how much this thick line will go around the cell. A jagged line to represent the irregularity of the cell border, and a half ellipse on the tip to represent the protrusions. Now, this what we also gain, that we are representing multiple dimensions by combining different visual encodings. So using Phenoplot, you can see here now a representation of breast cancer cells that we grouped based on image features and shape features into 5 clusters. And now you see directly when you compare with the heat map, now we have much more visual picture, a quantitative pictorial picture of how these cells look like. These cells in cluster 1 are small, these are bigger, and they are bigger, but this one has higher cell adhesion, this one is more irregular in the border. So this is help us and help biologists to understand the data better. However, a Phenoplot has many limitations, so it was aimed at cell shape and assuming that the cells look like an ellipse, and also the arrangement of the ellipses was fixed. It was written in MATLAB, and there was limitation in the visual elements. So I really wanted to take these ideas further and make a generalizable tool that can apply to many more data sets. So now I created a tool called ShapoGraphy. It is now a web-based app, so it's much more flexible and also much more fun to play with. We have some demos, and basically the user can create any structure they want by combination of shapes, and then start mapping their numerical data to these shapes. So the user can start from existing shape or even draw their own. So like a cell, like a star, or they're representing some information about fish or a liver, etc. Whatever they are measuring, they can draw something that look like it. But the real power of ShapoGraphy, and even I underestimated that, is when we combine shapes together. And this is really give us now a lot of flexibility to create complex structures that can represent a wide range of biological imaging data. So for example, you can draw a cell shape still, but you can represent even a well with a wound in it, or a flower, or a tree, etc. And for each of the created shape, you can map many variables, because each of this we have defined set of visual elements. So the easiest one is the dimension. So what the dimension of the shape, the border overlay, so how much it goes around the object border, or how much the symbols will go around the object border. The same filling with the symbols, or color, or spikes. For each of the elements, we have different options can be customized. So for example, we introduced the mesh element, and you can see here there is different configuration. And basically what this element do is, based on the variable value, it will increase the density of the lines. So these can represent the fiber density or something like that. So I want to give demo in this talk, but please come to the poster to see how it works. Simply, all you need to start with is an Excel file. You have the row names in the first column, and then you have the numerical variables. You load your data, you add objects, and then for each of these objects, you have the visual elements I mentioned, and you define for each of those which feature you want to use. You also have the option to plot your glyphs in a scatter plot, which give you a way to sort your data. So the first example I will give is on wound healing data. So in this assay, we introduce an artificial wound and observe over time how much the wound will close. So what we're measuring here is the density of the cells and how the wound area, how it will change. And you can see in the bar chart how this looks like. So using ShapoGraphy, I created a square to represent the well, and the density of the cells is represented by the color. So the higher the color, the darker the color, the more cells there are. I created a rectangle to represent the wound height. And the same information, cell density, can be represented by multiple visual elements. So here I'm adding it for comparison. So the more dense the mesh here, the more cells there are. And using just two variables, looking at it using ShapoGraphy, give you a completely different picture that is much, much more intuitive to look at. So we can see straight away that actually these two genes depletion result in a similar wound area, but actually when you look at the cell density is very different. And you can start also seeing the relationship between the variables. Another example is from this paper that was published by Belkman's group using multiplexing technologies. So basically looking at 40 proteins in the same cell simultaneously. And as you can see, these proteins have very different localization patterns. So this transcription factor in the nucleus, these genes, proteins are in the endosomes, while this nuclear pore protein in the membrane of the nucleus. So I have this data for 500 HeLa cells, and I plotted it using UMAP and done the clustering, which very nice. It showed me the overall structure of the data, but it is really difficult to know what these clusters means in terms of the biology. So looking at a heat map is can help to look one by one, but it's not intuitive. And it's also still rely on us building these relationships that in terms of the structure of the cell. So using ShapoGraphy, I used different combination of objects to represent different signaling activities, the cell context and organelle information. So for example, here I draw this custom shape for the cells. And then to represent the phosphorylation of AKT, I use the visual element border symbol, which you can see like how much it will go around the cell will reflect the amount of this protein. I added a square for cell context, and the density of the mesh is mapped to the cell density. And by adding more objects, layering more objects, I represented additional proteins. So for here, I'm representing different proteins in the mTOR signaling pathway. And then I added circle for the nucleus, and the color representing the level of YAP transcription factor. Added more information by looking at the proliferation, as well as how much of this nuclear pore protein that localized to the membrane based on the border symbol. Also, other organelles can be represented by color gradient, and as well as the endosomes abundance based on simple filling. By computing the average for each cluster and position these glyphs in dimensional space based on the cluster position in UMAP representation, we can see very nicely now that these three clusters have high cell density, and these have lower cell density. You can see high level of mTOR signaling in cluster 3 and high transcription in cluster 5. So compared to heat maps, and it's still very useful to keep the heat maps on the side, but it is give us a really different picture. It helps our brain to process this information and make relationships between these variables. So what important about the ShapoGraphy, that actually once you created these structures, you can save your work. And this is very important because some of these structures are complex, and we're trying to define different templates to suit different use cases. So there are demos on the website, and you can edit them, you can hide some elements or delete some objects, add new objects, etc. One challenge that we still trying to think how to overcome is actually that because we're giving a lot of flexibility, that some variables, some elements will interact with each other, and we still it can be very challenging to assess such interaction. So in summary, ShapoGraphy allow effective data visualization, and the aim is really to facilitate the interpretation of multidimensional phenotypic data. But importantly, to also give the user the power to create a new visual representation. And to my knowledge, there are no any other method that allow creating glyph-based representation in such way. It's applicable to a wide range of data, and it's very important that actually you can save what you created. So once you created a structure or a specific mapping, you can save it and download it as download the template, and it will be a JSON file. You can then upload it again with your data. We are creating a library of templates, so we hope that we can try to have a specific template for different types of data. So we would love to hear from you if you have any specific examples or some interesting data. One important thing as well is when we abstract our visualization in this way, it can be easier for wider audience. So someone who is not expert in microscopy might find it much easier to look at this visualization and say, I now understand what you mean, because looking at the raw data is really difficult. So at the end, I would like to thank my funders, the Wellcome Trust, for funding my research, and the people who done the work, Muhammad and Yuon, and my collaborators. The website is available in shapography.com, and please get in touch, an email on Twitter if you have any questions or you need any assistance with the tool. And please visit my poster to see more demos. Thank you for listening, and I'm happy to have any questions.